Well, good morning, everybody, and welcome this morning. I hope that you're all doing amazing. And
That was a beautiful start. I'm going to have you hang on just 1 second.
good. You give me the thumbs up.
I will. The joys of a virtual conference. All right, take it away.
Well, good morning, everybody, and welcome this morning. I hope that you're doing great and looking forward to not just a great session, but a great weekend. Hey, as we start, I'm reminded of a story. Have you guys ever heard of the two hunters and the grizzly bear? I heard it this past week, and it goes something like this. There's two hunters out in the woods, and they come in contact with a giant grizzly bear. And they're frozen in time, looking at each other, not knowing what to do. And one hunter goes to the next and says, "What do we do?" The other one says, "We should run." And the other hunter returns and says, "I don't think we can outrun this bear." And his other friend, with all his wisdom, says, "Well, I don't have to outrun the grizzly bear. I just have to outrun you." And that's just a little bit of light humor for you this morning. But you know what? It got me thinking. Running. Are you joining us this morning running from this past year? Are you running from maybe some challenges that the pandemic brought, or running from maybe even the challenges that PFIC itself is bringing to you? Well, here we are to the Family PFIC Conference 2021. And I hope you slow down for a moment and come together, join us as we're about to network, connect, we're about to learn together. Hopefully we can contact each other and encourage and maybe even give each other some hope. So stop running and just hold up a minute and just join us. My name is Walter Perez, and I have the privilege of sitting on the PFIC board and the privilege of being the president of the board. I live in Edmonton, Alberta, Canada, and I have a 4-year-old child, Armando, who actually has PFIC 2. And that's what brings me into this journey, joining the PFIC network. And I'm excited. I'm excited that you have chosen to spend a couple of days with us, and just maybe just checking us out, or maybe there's a session out there that is really catching your attention. Because you know what, the PFIC network, it is doing things right now. It is exciting to see where we're at. If you look at this conference itself, it's been a couple of years since we've got together, and regardless of maybe some challenges that have taken place, we've been able to come together virtually and bring it to you. And so I'm glad that you stopped by, because uh, you're going to get a glimpse of what is going on in the network, whether that's checking out our website, which is, I believe, a state of the art website with a lot of resources and information that I believe is evolving not only daily, but weekly and monthly. So there's always something going on there. It's come a long way. Whether you're looking at resources or even some of the committees that are involved, like the the parent advisory board that meets on a monthly basis, or we have a medical board and a medical advisory board that has a huge contribution, and even um, as we look at some future things that are evolving, whether it's the global PFIC committee, which brings global leaders together to not just encourage or share resources, but just to stay connected on a global level. There is so much going right now for the PFIC network, and I'm glad that you're here to be part of it. I'm reminded of a quote that says, "If you want to go far, go alone." Or sorry, if you want to go fast, go alone. But if you want to go far, go together. And this is a prime example of what's going on in the PFIC network. Together, we are doing so much. There's groups of volunteers and group of people that are coming together to make this happen. And so for that, I'm reminded of specific 3 individuals that have been a huge part of the PFIC network. We have Melanie, Tara, and Emily, who are co-founders and came together with vision. And you're about to hear from 2 individuals, uh, Emily, our executive director, and Tara, who's our vice president of the board. And let me pause for a moment and say, if you have not had a chance to meet them or had a chance to connect with them, do so, because you're going to be surprised and you're going to be impacted by what they do, but just their heart and vision for you, families that are coming in contact with the PFIC network. They just have they're amazing leaders, and just want to add value. And so I'm very thankful that I have the privilege of working alongside them. And please get around them, listen to their hearts. And without further ado, let me introduce to you Tara, as she'll share for a moment. Tara, over to you.
Thank you so much, Walter. That was really kind of you to say. Thank you for getting us started this morning. And I just welcome everybody to the PFIC conference. Thank you so much for taking time out of your day to join us. I'm so excited to get started. And Walter really, he really hit on it very well. That we have just come such a long way. So my name's Tara Kearns, and like he said, I'm the vice president of the board and one of the co-founders. It's so wild to hear where we are today. Just a few short years ago, Emily, Melanie, and I were, meeting virtually for the very first time. We met in what started as a Facebook group. Some of you patients and caregivers are part of that Facebook group. And at the time there was a website. That's all there was, pfic.org. And it hadn't been updated in a while. And, it was started by a mom who just wanted to get information out. And she was reaching a time in her life where she wasn't able to upkeep it and wanted to hand it over. So Melanie, Emily, and I all volunteered. We were all strangers to each other. And one of the very first times we met on Facebook Messenger. We realized that there was a chat feature on Facebook Messenger, and that's the very first time that we met. So it feels like a lifetime ago, but really it was about 5 years ago. And up until that time, I had never got to meet face-to-face with another family. I had never met another child that had PFIC. And I can't tell you the impact and the power that those first moments brought. It was just so moving. And so in the early days, Melanie, Emily, and I, we would have, we can't even call them board meetings. They were really times where we would get together and we would come up with ideas, right? We would talk about our struggles through PFIC. We would talk about our dreams for PFIC. And we said, let's just rehab this website. Let's just do it. Let's update the website. Let's get some more information out there. And that was really our goal, was to just help monitor the website. Well, for many of you other PFIC caregivers, you know that we can't do anything on a small level. Everything is constantly learning more and trying to grow and trying to understand. So we all have that in common. I think when it comes to our children, we're all deeply motivated for answers. So there were many nights that Melanie, Emily, and I talked about our dreams for PFIC. We wanted to build awareness. We wanted to become more than just a website that families could land on. We dreamed about networking and creating relationships and helping people find hope that we didn't feel like existed when our children were diagnosed. We wanted to connect to a medical community. We wanted to talk about gene therapy. We wanted to talk about drug trials and changing policy. We wanted to do so many things. And just 5 short years ago, it really felt like pipe dreams, right? But we were determined. So what started as a website quickly turned into an application for nonprofit status. And that's how the PFIC network was born. Just 3 moms trying to make a way and trying to make a voice and a name for this very rare disease. So that's where we started. And so it's so awesome to see how far we've come. 2 years ago at our first PFIC family conference, it was the first time that I met in person any other PFIC families. That's when we met Walter, who's now the president of our board. That's when I met Emily, who's our executive director, and many other PFIC ambassadors and board members. At the time, we were all just strangers, but very quickly realized that we had been lifetime friends and didn't even know it. So I just cannot thank you guys enough for joining us on this journey. We have already grown so much, and I can't wait to see where the next 5 years takes us. We all have passion and commitment, and we couldn't have done it without the leadership of Emily, who again was one of our co-founders and who has now been appointed as our executive director of the PFIC Network. So I would like to turn it over to Emily, and she's going to share some words with you and a welcome. And so thank you again, guys, for joining us.
Wow. Thanks, Tara, and thanks, Walter. That was an amazing introduction to you guys and our wonderful organization. My name is Emily Ventura. I have the privilege of serving as executive director for the PFIC Network. I have the privilege of being a part of the PFIC community as a parent and caregiver. And I have the privilege to know and meet members of the community worldwide, both patient, caregiver community, and the scientific community. I think that Walter and Tara outlined it beautifully. Our organization started small. It remains small. However, we have accomplished a lot. We have a fairly solid foundation in all areas of our mission, support, education, advocacy, and research. And we plan to continue to grow that foundation. We're going to continue to offer the same support services that are there and available for everybody. And we've learned to access, the amazing resources that are out there, translation services and, outreach, opportunities, sending educational materials to the wonderful providers that take care of our patients. We plan to continue to do those, and we plan to do more. As we move forward, I'm really excited, to say that we're planning to put together our very first research funding opportunity that should be coming out by the end of this year. This is a patient-led initiative, so we're working towards bringing the patient voice to research. The scientific community wants to know, they want to know what we need. So, our goal here going forward is to identify the needs of our community, bring those needs to the scientific community, and bridge that gap, find ways that we can work together to find those solutions. So please take our patient priorities in research survey. It's here on the platform. We hope to hear your voice. We hope to bring your voice to the scientific community. And without further ado, I would like to turn it over to the wonderful presenters who are going to start us off from Cincinnati Children's Medical Center. Just a few of the wonderful providers, researchers who are working hard towards finding answers for the PFIC community. Dr. Yin and Dr. Asai are going to start us off. Dr. Balistreri unfortunately had an emergency come up, so Dr. Asai is going to take his place as part of his team. Just a brief note before we get started, a few housekeeping things. If any of our members are using the translation software for the first time, you can look in the left-hand corner of your screen, top left-hand corner. There should be a button that says live. You can click that button and then you can go in, it'll open up another tab. On that tab is where you can select your language, and that'll set up a closed captioning service. It is AI based, so it is a translation software. It may not be perfect, but hopefully it'll help to kind of bridge that language gap. A few disclaimers. These webinars are set up for information and educational purposes only. They're not set up to give medical advice. If you have any questions specifically related to your case, please consult your medical team. Our Zoom is not a HIPAA compliant platform. So if you are asking questions, please leave all patient identifiers, names, and birthdays and such out. And this session is being recorded for future use. If the presenters give permission, we will be able to use them on our website, but they will be recorded and used in the platform and they'll be available for the next 2 weeks for the conference. If you have any questions, you can ask them throughout the chat, throughout the session in the chat, and we will address all the questions at the end of the session. Without further ado, I'm going to stop. And Dr. Asai, I'm going to turn it over to you. So thank you very much.
Do you hear me well?
Yes, loud and clear.
Okay, let me put this in screen mode. You see presentation?
Yes, we
Okay.
do.
So thank you everyone giving me this opportunity. Unfortunately, Dr. Balistreri having emergency, and he asked me to present his slide set last night. So he wanted to apologize for missing this opportunity, but I'm do my best to present this slide, wonderful slide from Dr. Balistreri. My name is Akihiro Asai, Asai. I'm a partner of Dr. Balistreri, one of the pediatric liver doctor in Cincinnati Children's Hospital. And we work together and we share some patients, and we see liver patient in our hospital together. So today, we have 3 big pictures, goals for this talk. One is this: put the PFIC and other genetic disease in the history of medicine, because Dr. Balistreri has a long history and experience, and he wanted to put how this disease was conceptualized and what's the progress we are making for this disease. And to understand PFIC, we will need to learn about the bile acid, and hepatocyte, which is the liver cells. And also, we want to put some new progression about this 2021 version of diagnostic process for neonatal cholestasis. So for those 3 topics, I hope to cover this talk. So we start with this history. In the past, about 40 years ago, we had named this disease named neonatal cholestasis. This is very vague name. It's mainly saying a newborn baby has jaundice, nothing else, because we did not understand any mechanism into it. So back then, we had, when the baby was born with the jaundice, abnormal jaundice, not the breast milk jaundice, it's the persistent disease jaundice, we only had 4 possibilities. One is this neonatal hepatitis, which is unknown, it's just having that. And the biliary atresia, that's the very different disease category. That's actually devastating disease also. And others are miscellaneous, and the others are virus, which is the infection through pregnancy from the mom. So majority of the disease we did not know, so they just put the name neonatal cholestasis. So this since 1970, it's been a long years to figure this out. So back then we are calling neonatal hepatitis neonatal cholestasis, or giant cell hepatitis, because when we do a biopsy for those babies, we see, sorry, it's behind this comments, but there's a big hepatocyte, the liver cell, that's what we call giant cell, with inflammation around. And we did not know what's going on. We did not know the cause. So we calling like idiopathic or sporadic. And sometimes it clusters in the same family. So we calling familial. But basically, we're just describing what's going on. We did not know anything about why this disease is happening. So this is Dr. B's comment that beginning the wisdom, call things by the right name. So we have to really put the right name to put our thought into the focus. So back then, 1978, the expert at that time from the worldwide get together and they're writing this article. They're trying to put the right name for this disease instead of neonatal hepatitis. So we put, at the time, we were trying to coin the name, and I think it was the consensus at the time, they started using the term for PFIC. So there is some background for this. Back in 1977, it was, we were calling progressive familial intrahepatic cholestasis. So we try to describe as detailed as possible to put the name into it. So this was the beginning of name for the PFIC. So kind of fast forward. So bottom line right now, what we know is we know that's a genetic disease and that's the gene defect. It's causing the liver disease. The liver disease is the intrahepatic cholestasis, meaning that nothing wrong outside the liver. It's within the liver that causing the cholestasis. And we are now, what we are doing right now is each gene makes molecule in the hepatocyte, and those molecule has some function. And if you lose those function, you have disease. So we kind of trying to connect all the like molecular level of finding to the disease. So that's now, this is current. So this now 2021, when we have a baby with jaundice, disease jaundice, we have this many possible disease. So at the time it was 65, but now it get to the 25%. The others, we are finding the cause of it, cause of those other disease. So the big pie, 65% of those unknown disease is now 25%. And with this 25, we are start identifying PFIC, Alagille syndrome, and bile acid synthetic defect. Those are all related to the bile acid metabolism or bile acid pathway. So we are making the progress in terms of identifying the disease. And within this 25%, PFIC is one of them. So this is actually one of the histological comment back in 1930. This Dr. Rich was commenting, there is a disease that he did not know anything about PFIC, but he was predicting there's a disease like a PFIC exists in the babies, because he was saying there is a jaundice baby without any obstruction of bile flow. So it must be a process of making the bile and then excreting the bile out of the liver. So this was his prediction about the PFIC. So let's move on. To understand the PFIC, we will have to learn about bile and the bile acid and the liver cells. So let's start with this quickly. Later on, Dr. In and I think Dr. Thompson will go over those details. I'm just giving the big picture for now. So this is when you cut the small piece of liver and put it in a microscope, this is what we see. So those are the anatomy, like a microscopic anatomy of the liver. And those red ones are arteries, blue is vein, it's a portal vein, so it's coming from the intestine to the liver. And this is green one is the bile duct. So the blood runs through this white area and it goes draining to this central blue part. This is the draining vein. And then between those irrigation system, this is the pink one. Those are the cause, or we call the liver cell parenchyma. So basically the blood coming from the heart, coming from the intestine, wash through the liver between those sinusoid space and give a nutrition, give oxygen, and take out all the waste, and then goes to the draining vein. And then if you look close, what's going on in this green part? So those are the one liver cell we call hepatocyte, and this one makes bile. This one produces bile and then dumping into this green area. that we call bile canalicula. It's a small, tiny draining pipe between the cells. And those green tiny tubes get together and then make a bile duct. And that's the dumping mechanism of those waste or bile goes to the intestine. So just more close up. This is like a high magnification pictures. This one hepatocyte and the second hepatocyte facing together, and they make bile acid. This bile acid is important ingredient of bile. If you don't have bile acid, you cannot really make bile. So bile acid is the most important component of the bile, which is made from cholesterol within the hepatocyte. So the hepatocyte, the liver cell, makes cholesterol, which makes bile acid from cholesterol. And then this bile acid needs to excrete, meaning that get out the hepatocyte and then goes into bile canaliculus, which is the draining small tiny tube, and then they get collected and make a flow to the intestine. So after you get this bile into intestine, they run through the intestine and then get absorbed back to the hepatocyte. So they have a circulation from hepatocyte to the intestine and intestine to the hepatocyte. So this is called enterohepatic circulation. So what is bile? I think it's, we're not gonna go deep detail about it, but it's made from cholesterol. So it's a biochemical ingredient your liver makes. And with this bile acid, your liver makes bile flow. Without bile acid, you cannot really have a flow. So there is actually two bile acid. One is a good one. Most of the bile acids are a good one for you. There is some bad ones. I'm just oversimplifying it, but this is probably important to see the balance between them. So if you want to have a more good bile acid in your body and in your intestine, and then less bad bile acid, you need a balance for this. So what can go wrong in this system? Well, it's a lot of steps. So each step has the error because each step will processed by the protein that your gene makes. So if you have problem in the genes, you cannot make those proteins and enzymes. So each step of those could go wrong. And it actually is going wrong in the patient with a PFIC. So I'm gonna go quick for this. There is each step, so you have to make bile acid, but if you can't, you have a disease called these disorders of bile acid sensitivity defect. If you have a defect in terms of excreting those bile acid, you have a transport issue. And you have, if you cannot really make those small tiny bile ducts, you have a problem. That's the Alagille syndrome. And you have other concerns that sometimes medication, sometimes infection can do the same thing. So this, I think we can skip this. So there is each step of this bile synthesis and excretion. There is each gene for each step. And Dr. Ying will go over for those later on, but I'm kind of fast forward. So if you make bile acid, it has to get out. And then to do that, you need to have this gene called BSEP. And to BSEP to function, you need to have this gene called FIC1 and IMGO3. So you need those three genes functioning very well in the canalicular membrane for the bile acid to get out. And then when it comes back, you need to have those genes to uptake. So each step has a possible mutations and problem. So as we know, PFIC1 is this FIC1 made by this gene. If you have a mutation in this gene, you can have PFIC1. Same thing with the PFIC2, same thing with the PFIC3. Those detail will come later in the Dr. Ying's presentation. So also, we are still seeing that not just those process of bile acid, you have to have a tight seal of this bile canalicula. So you need to have a tight junction here between the cell. If you have a mutation of these proteins, you have a not so tight junction, so you can have a leak of those bile acid or bile leak between the cells and goes into the blood. That's caused the problem. So this is Dr. B's picture that this story, and going back to the history. So it's starting with these 3 genes that causing PFIC 1, 2, 3. Now this research progress, we start seeing more and more genes involved in this disease, and it goes to a certain categories. So that's why he's making these branch pictures. I'm gonna go quick over it. So there's another gene called FXR related to those genes. And there is other set of genes that that is important for the bile acid metabolism. And if you have a mutation, you have the PFIC, and you have a other group of genes that's important for vesicle trafficking. And if you miss those genes, you can have a PFIC. And there is some more new genes that keep getting found and causing the disease. So I think this list of genes will go more and more as our research progress. So in this setting of those new disease and new genes coming up, how are we making a diagnosis? So this is 2021 version of in the clinic, how to make that diagnosis. So in this situation, first we start with patient. So we see the patient. We found cholestasis, not the usual jaundice, it's disease cholestasis. And then we run the blood test. This biochemical assay is basically the blood test. And we start seeing more and more concern for PFIC, and then we do liver biopsy still. It's important to see the actual event going on in the liver. So we do a biopsy, and then we stain some of those proteins that what we talked before. We have this technique called immunohistochemistry to identify those genes in the liver. And then now we have those molecular genetic testing. We have this is new, and then it's getting more and more sophisticated and more available and faster. So we integrate those genetic testing actually early in the phase. So this is the comparison from old to new diagnosis method. So the old, we were doing the biopsies and tests and everything, and at the end we do genetic tests to confirm. If it's correct, then good. If it's not, then we don't have anything to do. But now, because we have a next generation sequencing technology, which is basically much faster, much efficient genetic sequencing test, we run this genetic test early phase of the diagnosis process. So we are now calling sequence first. So at this point, what's doing it in this sequencing is basically we are reading the DNA patterns. So DNA has 4 different patterns and like letters in the DNA. Then we read each one of those sequencing, and then we target for those genes that are related, and then trying to find out. Or if nowadays we can just read all the genes at one time, which is the whole exome or whole genome sequencing. That's we just basically screen everything out at one time. For this diagnostic genetic part, Dr. Ying will go in detail a little bit later. So for those new approach of 2021, what we can do, so we can actually find the genes that's causing the disease. That's the main purpose of this disease. But at the same time, we're now finding a new type of mutation, which is the VOUS, the variant of unknown significance, meaning that it's new. No one see this type of mutation. No one really put into a context of clinical disease, but it's there. So it's new, and we have to do more investigation for those. And also, we have completely new genes that are not related to the bile acid metabolism, but now patient has the PFIC, and then we found this mutation in this new gene. Maybe this gene is important for the bile acid metabolism. So those are the ones we are working on. So that's the experimental model system to finding out what's happening in the patient, and the information we got from gene sequencing, we are connecting those two together by the research. So we have this test In the past, it was 25, 27 genes, but now we have a 66 gene to sequence at one time. And in the past, it was this EGL genetics, but now they have a shift in the business model. So now we are sending this to a prevention genetics. That's the company. If you need more information about this test, this is the, you can type basically the prevention genetics and then cholestasis gene panel. That's what we kind of run. So those are the one they will test in this company. And then the red ones are the one we just quickly talked about. So there is some more and more genes in this list. And then more to come, actually. Those are the new genes that are identified in the past 3 years. And those are the ones will be integrated to those tests soon. So this is kind of overview of what happened in the past and we are doing right now. And then this will we will continue to improve making the diagnosis, trying to connect the new finding and trying to understand more the bile acid metabolism so that we can understand more what's going on in the baby patient. And hopefully we can find the treatment options, better treatment options in the future. So this is basically, first we make the diagnosis. So Dr. B always says every patient deserves a diagnosis. And then this will allow us to go to a personal approach because each one has a different type of mutation. And based on the mutation, we will have a better functional classification of those mutations. And we will be able to tailor treatment and hopefully cure for those patients in the future. Thank you very much. I'm handing this to Dr. In.
Right. So thank you, Aki, for the talk. And then I will just continue with this topic and fill some holes you left and then add some explanation to the genetic testing in specific. So thank you. All right. Okay, so let me start the presentation right here. So hi, everybody. My name is Chunyue. I am a researcher, assistant professor working in the Division of Gastroenterology, Hepatology, and Nutrition at Cincinnati Children's Hospital. My own research is to study the mechanism of PFIC disease, mainly using animal models. And the goal is to develop new and better diagnostic and therapeutic strategies. I'm mainly focusing on PFIC2. I also work with clinicians like Aki and Alex and Dr. B, and also human geneticists to work on identifying new genes and new mutations that cause PFIC. So today I am going to give a brief introduction of the genetic testing of PFIC. And then I will talk about how genetics works and what genetic testing results mean, and then what the testing can and cannot do, and then the challenges we are facing. So this is kind of a biology 101. We all know our body is made of trillions of cells, and each cell contains a complete set of a person's genetic information. It is the DNA that is packed in these thread-like structures called chromosome. The DNA contains the instructions that guide how a cell should function, and it is encoded within a sequence of units represented by the letters you see here, the A, T, G, C. And these units are called bases. Sections of the DNA are called genes. And then the genes make proteins to help the cells to carry out their functions. And then the basic unit of the proteins are called amino acids. So next, oops. So when there are changes in the DNA sequence of single genes, you have mutations. And PFIC is actually a group of childhood cholestatic liver disease with similar clinical features, but each PFIC type is caused by mutations in different genes. I am listing the most common type of PFIC here, and I will go into a little bit the mechanism here. And then I know Dr. Richard Thompson is going to go much depth than today's my talk. So as Aki talked about, like this is a diagram of how hepatocyte cells look like. You have two hepatocytes side by side, and then in the middle you have this structure called bile caniculus, and this is where the bile fluids get excreted from the hepatocyte into the bile ducts. And this excretion process is regulated by a bunch of bile acid transporters on the caniculus. So you have this PFIC1 protein that is important for the structure of bile caniculus. And then this ABCB11 gene encodes bile salt export pump. So it is the actual pump that kick out the bile acid. And then this ABCB4 gene encodes a protein called MDR3. So as Aki mentioned, some of our bile acids are actually toxic, and you don't want it to just flow around in your bloodstream, cause damage. So the function of MDR3 protein is to pack the bile acids in a structure called micelle, so that it is protected or separate from the outside world. So if you don't have this protein, then the toxic bile acid is gonna go everywhere and then damage cells. So if you have mutations in these three transporters, obviously there will be liver damage and there will be problem in the bile excretion from the hepatocytes. Aki also mentioned the tight junction. So it is the function of the tight junction is to seal off the bile caniculus so the bile will not leak out. So if you have mutations in proteins that make the tight junction, then you will have leaky tight junction. So the bile may go into the bloodstream and then to places that it's not supposed to be. So mutations in this tight junction protein 2 is what we have here for PFIC4. And then more recently, there are other proteins and mutations discovered to be connected to PFIC. So FXR is a protein that regulates the expression of these transporters. So if you have bad FXR protein, then these transporters are not expressed at the correct level, so you have problem. And then this is PFIC5. So myl5b is a protein regulate where these transporters are located. So if there's a mutation in myl5b, these transporters will not get to the bile caniculus, so they cannot function properly. And then this is what we call PFIC6. So, so the mutations we found in PFIC patients, they got different. And there are two main classes. One is called point mutation. This means there's a change in one single base in the DNA. So I put an example here. Let's say a normal DNA sequence is ATGC, and then for this point mutation, this G changed to an A. The second big group is called frame shift mutation. It is mainly caused by either the loss or gain of bases. So again, normal is ATGC. In this deletion mutation, you have T and G deleted. And the insertion, you have T and C inserted in between T and G in a normal sequence. And then duplication is you have a, it's pretty straightforward, it's a duplication of the normal sequence. So the consequence of these different mutations can vary too. So I put an example here. This is the protein of bile salt export pump, the protein that is impaired in PFIC2. So you can see it has very complicated structures and different components. So this is what a normal BSEP protein should look like. Some of the mutations that they actually do not cause any change to the protein itself. So we call them silent mutations. So they are harmless, they are fine. And then some mutations actually result in a much shorter protein, or sometimes it doesn't even make any protein. And this time you can see like this protein, mutant protein, stop right here. So it loses all these complicated structures that should be present in normal protein. So this type we call nonsense. This is probably the most severe type because you just completely lose the protein there. And then there's another group is called missense mutation. So it has changed the protein a little bit. Sometimes it's just 1 or 2 amino acids. So that's, that can, if the amino acid is so important, it is the key for the protein function, then it's very severe. But sometimes it may just impair the protein a little bit. So the protein still has some function. It's just not at the same level as the normal protein. In human, we have 23 pairs of chromosomes. So all the chromosomes are in pairs because one is from the dad, and the other is from mom. Likewise, we have 2 copies of each gene, one from dad and one from mom. And this is really important when we think about the genetics of PFIC. Usually, PFIC disease is actually caused by we call autosomal recessive inheritance. That means you have to have both copies of the gene defective to develop a disease. So when we look at this diagram here, in PFIC family, both parents, dad and mom, are usually carriers, which means they have 1 copy of the normal gene and then 1 copy of the mutant gene or defective gene. And then their children, 25% of chance you will have a child that who is completely normal. He or she doesn't inherit any of the affected gene. And then 50% of the chance they will be carriers just like the parents. So they have 1 normal gene and then 1 copy of the affected genes. So they are usually healthy. They don't have liver disease, but they will pass on the mutation to their next generations. And then unfortunately, 25% chance the child will have 2 copies of the affected genes, and then they will develop PFIC. So I also want to emphasize it's important to keep in mind that both parents are carrier. They each carry 1 copy of the affected gene, but the exact mutation in the affected gene can be the same or can be different. So let's see how this works. Now I am just going to talk about what genetic testing is. It's a type of test that identifies changes in genes, sometimes in chromosomes and the proteins. And then the results of a genetic test can confirm or rule out a suspected genetic condition, and then can help determine if a person's chance of developing a disease or passing on a genetic disorder. Like I said, because different PFIC types, they share some similar clinical features. The benefit of doing a genetic testing is we can figure out exactly which type of PFIC the patient has. And because the different proteins are affected in different type of PFIC, it is beneficial to know exactly which protein is affected, and we can develop the disease-specific type of treatment for the patients. So as Aki said, there are two types of genetic testing. One is called DNA sequencing panel or gene panel. So what these companies and hospital will do is collect the DNA from patients and then just sequence specific genes, and then see these genes are known to cause PFIC or other liver disease. And then if you find mutations in these genes, okay, the patient likely have that disease. But when the test does not find any mutations in the node genes, it will be beneficial to do a whole exome sequencing or whole genome sequencing. The difference is these whole exome, whole genome sequencing will actually sequence all the genes in the body, not just a few selected genes. So you get a much bigger comprehensive picture of what can go wrong in the DNA sequence, and then maybe we can get a diagnosis from this strategy. So I put a sample of genetic testing result here. If you get a positive result, great, you get some answer there. And then these are the components of the genetic testing results. So it will tell you which gene is mutated. So I put an example here. This is ABCB11. That's the gene that is mutated in PFIC2. So is listed here. And then AR here means this PFIC2 is autosomal recessive. And then it will list the actual nucleotide change. So in this case, C base at this position changed to a T. And then the consequence of this nucleotide change is a protein amino acid change. And then the zygosity says homozygous. That means they detect 2 copies of this same mutation in this patient. And then the type of mutation, this lab determined it's pathogenic, means it's likely to cause impaired protein function. So there's another possibility that the report may show 2 different mutations. That means the patient have 2 copies of mutant genes, but the exact mutations are different. That's why it's called heterozygous. So it has 1 copy of this mutation and then 1 copy of the other mutation, and both mutations are pathogenic. So overall, this patient doesn't have a normal ABCB11 gene. Both copies are defective. So this is a situation you can call, okay, this kid probably has PFIC2. Like I said, pathogenic mutations are the ones that have been previously reported in PFIC patients, and then its function has been studied and validated by researchers. So we know it severely impairs the function of the protein. And they are usually nonsense and missense mutations that result in complete loss or severe impairment of the protein. Sometimes you will see likely pathogenic, this term. That means based on the change in the amino acid, it is very likely that this mutation will impair the protein function, but it has not been previously reported or has not been firmly validated, studied by researchers. It is important to keep in mind that doctors cannot use just a positive genetic testing result to predict the course or severity of the disease. You still need liver biopsy or other lab tests to really evaluate the status of the disease. The reason why is there are many other factors that can affect the disease other than the mutations in these genes that are associated with PFIC. First of all is a person's genetic makeup. So you and I, although 99.9% of our DNAs are the same, but it is that 0.1% make us different human beings. And that 0.1% difference also will have an impact on, how your liver can tolerate the mutations and then how you respond to drug treatments. And also environmentally, what you eat, do you exercise a lot? Are you exposed to virus or toxins? Can have an impact of your disease severity and progression. That's why sometimes you have two patients with exactly the same mutations in PFIC genes, but they present very different liver injury. And also for just one person, you can have different stages of liver disease, and then it is because of the environment changes, or you have like life-changing events, like you get pregnant or you're under lots of stress. All these things can alter your liver disease presentation here. So that's why it is really important to still do the liver biopsy lab tests to monitor your disease. Then there's this frustrating part of genetic testing. So sometimes you get negative results. That means they cannot find any mutations in the known genes. But it is possible, just the technology is not good enough to detect all the genetic changes. So you need a further testing to confirm a negative result. So negative result doesn't mean you don't have the disease. I will put it that way. Unfortunately, 50% of patients with cholestasis do not have mutation in the known genes. So they do not have a molecular diagnosis. And then the third possibility is inconclusive results. So I still put this example here. If you look at the type of the mutation, it's called VOUS. That's variants of uncertain significance. And then in this case, this patient has 2 copies of this VOUS mutations. And also in other cases, you may have 1 copy of mutation that is pathogenic, the other is VOUS. All this cannot lead to a definitive positive diagnosis because VOUS is usually predicted to change amino acid, but it is not clear to what extent it will affect the protein function and to cause liver disease. So one thing that is helpful is to do genetic testing on your other family members, healthy or with liver disease. If VOUS is presented in all the affected members but absent in the unaffected members, it is likely disease causing. But this is all just kind of indirect evidence. We still need research to be done in the lab to study this VOUS and confirm it is actually disease causing. So with all these problems, the only way to kind of conquer it is to do research. So we working in the lab, try to develop new tools to diagnose and identify new genetic causes of PFIC. And then as we collect more different mutations and then the patient data, we can hopefully form a correlation like these type of mutation will cause more severe disease or less severe disease. And then the ultimate goal is to develop effective treatments so that we don't have to go through the transplantation route. So I think that's all I have on my slides. And then I will be happy to answer questions if you guys have any. And I can answer questions for Aki too. I will try my best. So, all right.
Awesome. Thank you so much, Dr. Yin. That was fantastic presentation. I can't get my video turned on at the moment. I may ping Jeff to try to work on that in the background. But until then, I'll keep talking to you. We do have some really good questions. A lot of that was amazing. I haven't heard much of that information. So really, thanks for breaking that down for us. A few questions about the genetics panel. So the first question we had was if a patient had testing performed through the EGL panel, should they also get the newer version, or should they also resubmit testing through the new panel?
Of course, if you get a result, you don't have to do that. And then actually, cost-wise, these gene panels are actually more expensive than the whole exome, whole genome sequencing. I first, of course, I will talk to the doctors and your genetic consultants, but I mean, if you do a panel of 66 genes and then the newer one is 88 genes, I think it's probably, for me, if I make the option, I will just go for the whole exome, whole genome sequencing.
Okay, so to clarify, the link that was in Dr. Asai's presentation, that's not for whole exome sequencing. That's just the genetics panel for those 66 genes that you have identified so far, but that that's not an all-inclusive. That's what's accessible now.
That's correct.
Great, okay. Could you actually very quickly give and you likely addressed it, but just to kind of hone in on this because it's kind of complex, a very quick comparison between whole exome sequencing and the genetics panel, just a quick breakdown between the two?
Okay, so the gene panels, they will only sequence whatever number of genes on that panel. So the newest version is 88 genes. These genes are all have been connected to liver disease, not just PFIC. So for the whole exome sequencing is you actually sequence every single gene in your body. So it's not 88 genes, it's actually millions of genes. And then there's also called whole genome sequencing. So in addition to sequence the genes themselves, they also sequence the part of the DNA that doesn't make a protein. So there are some we call like regulatory sequence exists in these DNA. They are important regulator of how much a gene should be expressed and where it should be expressed. So if you have mutations in this regulatory sequence, it will also affect the protein. So again, whole genome is the kind of the biggest, the broadest, because it really sequences every single nucleotide in the genome. So yeah.
Thank you for that. There's so many out there. It's very complicated, but we appreciate the breakdown.
Yeah, and then I was, because I work on these whole genome, whole exome sequencing results, very, like if you find pathogenic mutations in them, great, you get an answer. But if you don't, and then you find these genes, they have mutations and they can be important for liver function, but then you need to do research. Are they really the reason the patient developed liver disease? So that gets really, really complicated. That's why your lab cannot give you a definitive answer. That's usually the case. So
I see.
Yeah.
Okay. Another question from there's a few questions related to this topic. How common is whole genome sequencing as part of the diagnostic process? And is there still value for whole genome sequencing after diagnosis has been made through panel testing?
So I will say whole genome, whole exome sequencing for us researchers, it's probably more valuable because we want to discover new genes. And then that's the only way, like if we have a an overall picture of what's happening to your genome, we are more likely to identify things that are not reported before. But for patients, you need to find a good lab, not only just sequence your DNA, but also can like have connections to the hospital and geneticists to really have a way to interpret these genetic results. Otherwise, it's just giant data that is not very informative. So the whole genome, whole exome sequencing is only used when there's nothing found in the panel testing. So you go panel testing if you find something. So probably there's no need to do these whole genome, whole exome sequencing anymore.
Okay, so step one, panel sequencing.
Yeah.
And then if a diagnosis, you stop. If no diagnosis, whole genome sequencing.
Right.
And then hopefully something is identified.
Yeah.
If working with a laboratory that is trained to identify, correct?
Yeah. And also
Okay.
you want like reputable laboratory provide high quality sequencing because that can vary a lot. So
Okay.
yeah.
Okay, thank you. Another question from the audience, again related to this topic. So now that genetic testing is more readily available, is it recommended that siblings who maybe do not express PFIC disease, but could be carriers, or is it recommended that siblings get tested? And is that happening more often that you know of?
So if there's PFIC patient in the family and then that patient has a clear genetic diagnosis, then the healthy siblings can just sequence that particular gene to see if they have the same mutations, carry the same mutations. You don't have to do the whole genome exome sequencing. So that's kind of an overkill. Yeah.
Okay, but it would be relevant to kind of screen family
Right.
members.
Exactly. Yeah.
Okay, that was going to fall into one of my personal questions. If things have evolved to where we're identifying, or you're identifying if carriers of the disease are starting to show symptoms, or if that's a valuable part of the should that become standard for us families? Should we be thinking about ourselves and siblings as carriers? Have we shown a correlation between that yet in literature? Or
so far, I would say majority of the cases, PFIC carriers do not develop liver disease, but it's possible the environmental trigger, like pregnancy, or a big change in your lifestyle, what you eat, your metabolism level, or maybe you like take Tylenol or other like drugs that affect liver function, then your liver will react more to that than a normal, not carrier human being. So I will not overthinking it, but when you start notice you have liver issues and why, because you are a carrier, you are more susceptible to all these triggers than just like other people that do not have any mutations.
Okay. Well, that's helpful to hear too, from an accessibility standpoint. I know from my personal experience, I had a panel done just through research. This was 10 years ago, but it doesn't seem standard that family members get tested. And when we, our clinicians may decide that would be okay, but it's not necessarily covered. Testing isn't covered for family members yet. So I think that's something that's constantly on our minds, the accessibility. It seems to be more available.
Yeah, and then the cost is way down now. So yeah.
Okay. Well, that's good to know. So that's kind of hopeful for the future. Maybe we'll be able to access testing a bit,
Yeah.
bit more frequently. Thank you. Another question from the audience. So once a patient has a genetic diagnosis, how often should they have a biopsy?
So biopsy is invasive and it has its risk. So it's usually the standard is you keep track of your blood test results. And then if your AST, ALT, bilirubin levels go up and change a lot, then that's time to do a biopsy. But if they are stable, and you probably don't want to go through biopsy, and then now there are more advanced imaging technique. It can monitor fibrosis through imaging without, really going to the liver physically. So these are other options.
Okay. Would it be safe to say, once a patient has a diagnosis, the biopsy would be more so looked at to assess severity of the disease if there's signs of progression?
Yes,
okay.
That's correct. And then also, let's say you have a missense mutation, so you still make a protein, but if there's something wrong with it, sometimes you can get a biopsy and then do some, staining on them, and also just even look at the protein itself. You can get a sense of what is really the consequence of this mutation. And then that can help the physician understand the mechanism of the liver injury and then how to manage the patient. So it's helpful not just for diagnosis, but also for treatment information.
Okay, I see. Thank you. I have one last question before we wrap up. So I still hear the term idiopathic neonatal hepatitis. And I refer back to Dr. Asai's earlier presentation today. It seems that term, PFIC fell into that category, 30 years ago. But as the evolution of understanding, kind of moves forward, PFIC seems to is falling outside of that category. But is it still possible that a patient kind of would lump into that category, that idiopathic neonatal hepatitis, to not rule out PFIC as part of that?
So I can confirm with Aki later, but my understanding is that idiopathic neonatal hepatitis is the class that you don't have a clear molecular diagnosis. So if you have one, then you get out of that group and then you have your own disease type there.
Okay, but perhaps a patient who, they still potentially could have PFIC in one of the genes that maybe wasn't on the panel or whatever was sent off, that could still be in that category, just hasn't been identified yet.
That's right.
Okay. One last question before we go. If transplanted, do you no longer have the gene to pass on to your offspring years later, or is that still a possibility?
Unfortunately, transplant is just swapping out the liver. Your genetic information is still the same. You are still the carrier of the mutation. So, what you pass down to your offsprings is the genetic information in the germ cells, and then transplantation will not change anything to the germ cells. So you are still a carrier even after transplantation.
Okay, that makes a lot of sense. Thank you very much. And we are just at time, so I'm going to wrap us up. But if anybody has any lingering questions that they felt like they didn't get to ask, you can send us a message or hold on to them. I believe Dr. Yin and or a member of her team will be available in the panel Q&A tomorrow, so you can ask them then as well. The panel Q&A is at 2:15 tomorrow. So thank you very much for a wonderful presentation. I really appreciate both you and Dr. Asai joining us for the first one this morning. And our next presentation is in 15 minutes. So we'll see everybody then. Thanks, Dr. Yin.